Today I'm going to talk about the Genome Database for Vaccinium, or GDV, and how you can use it for genetics, genomics, and breeding research. GDV can be found at www.vaccinium.org and is a single website that has easy-to-use tools and searches. It can be tailored to the community needs thanks to the open source data platform called Triple, which has numerous modules available. As far as data, we curate genetics data from published manuscripts, and we curate genetic markers, genetic maps, QTLs, and MTLs. We have a tool for viewing genetic maps called MapViewer, which I'll cover later in this presentation. For genomics, we have genes and transcripts from Genomes and NCBI, and the genomes can be viewed in JBrowse, searched using the BLAST tool, and also viewed in Synteny Viewer and the Pathway Site tools, which I will talk about later. And for breeding, we have the Breeding Information Management System tool, which Dory Main will be presenting on later in this conference, and I will touch on it briefly towards the end of this presentation. The GDV homepage has a few key sections that I want to point out. The first is the header region, where we have various pull-down menus, and this is how you can access all the tools and data on the website. We do have a login prompt, but you only need an account for GDV if you have private data that has restricted access, or if you're using the BIMS tool. Everything else is available without an account. Down at the bottom we have a species quick start that will quickly take you to those crops. Those crops can also be accessed under the crop menu at the top. We also have a tools quick start, which again just has some quick links to the common tools that can also be accessed in the tool menu, or the toolbar menu. For searches, we have quite a few different data types. I'm going to talk about the more popular ones, and we have individual search interfaces for each of these data types. The first is the gene and transcript search, where you can search genomes and transcriptomes. We have a germplasm search, we have a map search, a marker search, and a QTL search. All of these are accessed via the search menu, and they can be customized to be very specific or more general. You can customize which columns are displayed in the results table, and you can download that results table. For the gene and transcript search, you can also download a FASTA file of the sequences that are on the table. And for genome sequences in that search, you can also specify flanking sequence links to add to the gene or mRNA sequences. We also have MegaSearch, which is a different type of search interface that has some different options and customizations. You start by selecting your data type. In this example, we chose the gene and transcript data type. You then can go and specify some details about your query, like you can specify a specific genome, even down to a scaffold and start and stop location. You then can also customize the downloadable fields. By default, everything is selected, but you can unselect all and choose which ones you really want. And you can also specify the file type by clicking the buttons up here. We also have a cross-site search function, and this is for searching across 3 databases currently. The first is GDV, the Genome Database for Resize, and the Citrus Genome Database. So I did a search of mRNA features for ubiquitin, and this is what I see for GDV, and here's an example of the GDR results. Basically, anything that's green is a hyperlink that you can click on and view the details either on GDV or on the sister website of GDR or CGD. So I want to talk about our MapViewer tool, and we have replaced CMap with this tool because this tool is much more cool. And it is a dynamic tool for viewing genetic maps. It allows the user to customize what they're seeing and the colors of the markers. There is options to export figures. Anytime you see a little camera icon, that means the figure can be exported, and you can use it in presentations or manuscripts. And this is accessed either through the map details page, which is what's shown here, or the tools menu. So if we want to see more details about a linkage group, we would just click on that linkage group. And what comes up is a view of an overall view of the linkage group on the left, and then a more detailed, refined view on the right. And the view on the right can be adjusted. You can resize the box and also drag the box to the region that you want and the view that you want. And when you hover over the gene and QTL names, it gives you some details about those features, and if you click on the map, the QTL or marker names, it will give you even more details. The map viewer below that, the diagram, you will see some options. The first section lets you change the reference map and linkage group that you're viewing. There's also a settings down below to change what's being displayed. So you can choose the colors of the markers, you can hide like all the SNP markers or SSR markers, and there's a few other options you can choose. And then in the middle is the select comparison map. When you toggle on the check mark here, it'll show a comparison map, and it'll show you all the maps that have corresponding markers, and it displays the number of corresponding markers for each map and for each linkage group. There's also an option to view the dot plot and the correspondence matrix for the comparison map. So here's a comparison map example. I just want to show that the corresponding markers have blue lines that link them together, and you can adjust the view, which region is being viewed independently on each map. And again, there is an option to export this when you have it just the way you want it. For genomes, we have quite a few genomes. We have some blueberry genomes, we have a bilberry genome, a few different cranberry genomes, and then some transcriptomes. The transcriptomes are aligned against the blueberry, all the genomes in JBrowse, and the transcriptomes are also available to search using BLAST. Most of these genomes have either been published or were contributed pre-publication by the authors. Some of them have been published and are now fully available. Some of them are in the process of being published, and so the data will become completely available as the publications are finished. We also have a Synteny Viewer. This is a viewer that displays data from a Synteny analysis we do as part of adding genomes to GDV. It is accessible from the Tools menu. Like I said, it displays analysis data from a comparison we did. It is pairwise comparisons between two genomes. To use it, you first select a genome and a chromosome or scaffold from that genome, and then you choose which genomes you would like to compare to. We still have some newer genomes to add. They'll be coming shortly, but it doesn't display all, like in this case 5 genomes, it just displays pairwise comparisons. So here's an example of chromosome 1 from cranberry compared against the bilberry genome, and you'll see all the different chromosomes from bilberry are around the circle, and as you hover over synthetic blocks they turn red and gives you a little information about that block. When you click on that block, you get a more detailed view of that synthetic region, and this map can actually be zoomed in and out of as well, and then it lists the corresponding genes here on the side. The green hyperlinks indicate that you can click on those and get more details about that mRNA feature. We also have a pathway psych tool. This is an analysis we run with pathway tools. This is an example of a cellular overview from a blueberry genome, and it shows you which biochemical pathways you can zoom in, you can click and see more details on this. I just want to point out that if you have metabolomics or transcriptomics data, you can overlay that data on here and it will actually give a heat map coloring scheme to the pathways that are represented in your omics data. We also have an expression heat map tool for if you have RNA expression data. We have a few studies uploaded to the database already, and you can view expression data on feature pages for the genome that the expression data was aligned to. And you can also generate heat maps using the expression heat map tool under the tools menu, where you can select an organism, you can search by function or go term, you add the feature names that come up to this box and hit display feature map or expression heat map, and you get this nice heat map where you can click on the treatments and the mRNA names and get more information. And there is an option to export this as a figure as well. So just want to point out, we do have a mailing list and contact form. If you would like to join our mailing list, it can either be found under the general section of the toolbar or at this URL. We have a newsletter, some how-to tips and webinar announcements that will be coming out on that mailing list. We also have a contact form, so if you have any suggestions or questions, please contact us. BIMS I'm just going to touch on briefly. This is a program to manage your private breeding program. Your data stays private, and you control who has access to it. If you want to learn more about it, please watch Dory's presentation later in the conference, or go to www.breedwithbims.org. And with that, I'd just like to thank our funding sources. Thank you.